So continuing with genetic recombination in bacteria and viruses. So there are three types by which genetic recombination can be done: conjugation, transformation, and transduction. In this video, I'll be talking about transduction. Now, transduction can be of two types. One is generalized, another one is specialized. So what is transduction? Transduction is an event in which bacteriophages function as vector or vehicles to transfer DNA from donor bacteria to recipient bacteria. Okay, so with the help of the diagram, we'll understand exactly what happens. Now, first I've drawn a donor bacteria. Now, within the donor bacteria, the circular chromosome is actually its own bacterial chromosome. Okay, so this is bacterial chromosome. Now, what happens? This bacterial chromosome will it gets broken up. Okay, and then it gets taken up by the bacteriophage. So now the bacteriophage is taking up this broken portion of the bacterial DNA. Okay. After that, this bacteriophage gives it to another cell, which is the recipient bacteria, and now the recipient bacteria will receive this DNA through a bacteriophage. Okay. Now, since this bacteriophage is helping in the process of transduction, therefore we call it as a transducing phage. Okay, it's a vector because it's helping in carrying, so therefore it is called as a vector. Okay. Now, what are the different parts within a bacteriophage? It has got a capsid head within where there is a nucleic acid or DNA, and then there is a collar and a tail. Okay, and it will help to take up DNA from a donor bacteria to a recipient bacteria. Okay, so now let's talk about the bacteriophage. Now, what are bacteriophages? Bacteriophages are actually viruses that helps to infect a bacterium, and within the bacterium, it also replicates its own DNA. Okay. Now there are of two types. One is temperate bacteriophage, and another one is virulent bacteriophage. Now, in temperate bacteriophage, we get to see lysogenic cycle, and in virulent one, we get to see lytic cycle. Okay. And what are the difference? In temperate, it does not cause disruptive infection. Okay? Instead, phage DNA incorporate into bacterial DNA and replicate lysogenically and eventually become virulent. And in case of virulent bacteria, they are capable of causing bacterial infection and eventually destruction and death of bacterial cell. Example is T4 phage. Okay, now let us understand the lytic cycle and the lysogenic cycle. Okay, this is the diagram of a lytic cycle. Now out here what's happening? From this particular cell, this is a cell which contains different portions of bacteriophage. Okay? That means it was previously infected by a bacteriophage and all the particles have been made within it. Okay, and the red portion is the DNA which is present within the bacteriophage. So what will happen after the infection? This cell will burst open and all a completely developed virulent bacteriophages will come out. So this is a virulent bacteriophage that has come out. Okay? Virulent phage. Now, this virulent phage will go and it will attack another bacterial cell and it will send its own DNA, push its own DNA within the bacterial cell. Then this DNA will get incorporated within the bacterial chromosome and therefore it will now look like this. Then what will happen? The DNA will replicate within the cell, and after replication, they will make their own replication happen. They'll make their own proteins and everything. They'll make their capsid and they'll finally develop all the different parts, and therefore the cycle goes on. This is the lytic cycle of how a bacteriophage infection occurs. Then now let's look at the lysogenic cycle. The lysogenic cycle, what happens after the infection has been done and the DNA has been incorporated. So what will happen? They will become double-stranded, and again the replication will take place. But even if the replication is taking place, even if the cell is going through several form of division, okay, even after division, the incorporated DNA is still present inside. So this is how lysogenic cycle take place. Okay, so there is a difference between lysogenic cycle and lytic cycle. Okay, now let us talk about the transduction. Now let's talk about generalized transduction. Now a DNA fragment is transferred from one bacterium to another by lytic phage, which carry donor bacterial DNA due to an error. Now virulent phage act as vector in generalized transduction. Now let's look at the different steps. Okay, a lytic bacteriophage. Okay, a lytic bacteriophage infects a susceptible bacteria. So here the susceptible bacteria is actually this particular bacteria, the susceptible bacteria. Okay, and this is the bacteriophage which is infecting, and the phage genome is being pushed within the cell. Okay, next, step 2. When phage genome enters the bacterium, it causes degradation of bacterial host DNA into fragments. So the bacterial host DNA, this was the host DNA which is getting degraded. Okay, okay, next. Phage genome replicates using host machinery and synthesize enzyme and code protein. So it has make, this is the code protein and the different parts. Okay, these are the parts, various parts, and this is the DNA, this is the DNA, this is the DNA. So they are doing their own thing and making their own parts. Okay, उसके बाद during maturation and packing of virus particles, few phage heads may envelop fragment of bacterial DNA by error that is double stranded. So only bacterial DNA, that is the host DNA, is present in the transducing phage. So what's happening out here? See, so this is the cell. So the DNA fragments of the host are there. Now instead of taking up its own DNA, it is not taking up the double-stranded DNA of the host, that is from the bacteria. So now it contains the DNA from the host cell, and now it has become a transducing phage. Now this transducing phage, what it will do, they will go and attack another cell. Okay, so they are going and attacking another recipient bacteria. So transducing phage carrying bacterial DNA host or donor will infect another cell and transfer the donor DNA to the recipient bacteria. So now this bacteria is receiving DNA from another bacteria, okay, because it is not the phage DNA anymore, it is actually the DNA from another bacteria. Okay, so when bacterial DNA of donor is introduced into the host cell, it can be integrated into the bacterial chromosome of the host and transfer several bacterial genes at one time, forming recombinant DNA. So now it has become recombinant because this particular DNA is just okay. So this DNA is coming from the host, okay, or I can say it is coming from the donor, and this the blue one is from the recipient. So bacteria recipient multiply with new genetic material. Now the genetic material has changed, and therefore these cells are now transduced bacteria. Okay, so now this bacteria have DNA from two different cells. Like the green one is actually coming from a different cell, right? And the blue one is from the parents. So only these that are located close together will be transduced together. So with the help of this, we can again understand the distance between the gene, if the genes are present close together, if they can be transduced together. So all this information can be taken up from here. Okay. Now, generalized transduction can be of two types, abortive transduction or complete transduction. Let's look at the difference between abortive transduction and complete transduction. Here you can see after the phage has given its DNA, it can go either of the two ways. In one, there can be homologous recombination, that is in complete transduction, where the DNA will be incorporated within the bacterial DNA, the one that we saw earlier. And in abortive, what will happen? Some part of it will combine with the endogenous, and this is called as abortive transduction. Okay. So in case of abortive transduction, the transient expression of one or more donor gene without formation of a recombinant progeny, the donor DNA fragment, that is exogenous, does not integrate with endogenous and also do not replicate. Only one bacterium contains exogenous and the other bacterium is like parent among the progeny. So out here we do not get everything. Like all the DNA is not incorporated and all the cells that are produced are not having proper transduction. So it is called as abortive. Okay, and the other one is production of stable recipient. From here we'll get stable recipient recombinants that inherit donor genes and retain the ability to express them. Frequency of abortive transduction is greater than complete transduction. So basically, complete transduction occurs very less, but this is more better for genetic recombination as compared to abortive transduction. Okay, so that's all for this topic. If you have any doubt, you can text me or you can call me.